All right, class, let's continue with our discussion on the adaptive immune system, also known as our third line of defense. Let's focus on how our lymphocytes, our T and B lymphocytes, are developed. During T cell maturation, we're going to have our T cells develop the receptors in our thymus gland and respond. This is going to be directed in response to hormones that are specifically located within the thymus gland. So T cell maturation is in the thymus gland. There are several receptors that I want you to focus on for the T cells. Those are CD3, CD4, and CD8 receptors. The CD3 receptors are going to surround our T cell receptors and assist in binding when an antigen-presenting cell presents an antigen to a T cell. The CD4 receptors are accessory receptor proteins that bind to our class II MHCs. And then finally, CD8 receptors are found only on cytotoxic T cells and bind to the MH1 molecules. Recall, MH2 molecules are present only on phagocytotically active molecules, and MHC1 molecules are present only on all nucleated cells in our bodies. So here's a stereotypical T cell, and our stereotypical T cell is going to have on its surface the class 1, the CD3 receptor, and that CD3 receptor is going to be associated with our antigen binding site, and then we have the CD4 receptor, which also aids in activation of our T cell. If we look at a B cell and its maturation, it's going to mature in bone marrow in humans. But it's not called the B cell because it's a bone marrow cell. It's named B cell because it was initially isolated from the bursa within chickens or within birds. And it occurs in humans in bone marrow. Specifically, it's going to occur in the bone marrow that harbors stromal cells. And these stromal cells are going to provide the necessary chemical signals that initiate the B cell development from our multipotent stem cells. Naive lymphocytes are lymphocytes that have not been fully developed yet. And they're going to circulate in the bloodstream and home in, so to speak, through chemotaxis to specific sites in our lymph nodes, spleen, and our gult. So here we can see a classic B cell. Our classic B cell is going to have an MHC class II marker on its surface to help it respond to antigen-presenting cells. And it's also going to have an antigen binding site as well on its surface. As we compare and contrast our B cells to our T cells, the B cells in humans are mature in the bone marrow versus T cells in the thymus. On the surface of our B cells, they're going to have immunoglobulins as their surface markers, whereas the T cells have our CD molecules. And we highlighted three specific categories of CD molecules, so cluster or differentiation molecules, earlier, a couple slides ago. We have low concentrations of T cells in the blood versus B cells in the blood versus high concentrations of T cells. Our B cell receptor for antigens are going to be immunoglobulins versus our T cell receptor, which is distinctly not an immunoglobulin. We have B cells located in our lymphatic cortexes, whereas the T cells are located inferior to our lymphatic follicles. Our B and T cells also differentiate between their need to have an MHC molecule present. So our B cells do not require an antigen to be presented with an MHC molecule, whereas the T cells do require an antigen presented with an MHC molecule for activation, with the exception of the gamma delta T cells. Those can be activated by a PAMP, or pattern-associated molecular pattern. Our B cells produce plasma and memory cells, whereas the T cells produce multiple kinds of T cell, multiple subtypes and memory cells. The helper T, cytotoxic T, the regulatory T, and the gamma delta T. Our B cells, generally speaking, are going to make antibodies to neutralize antigens, whereas the T cells are going to destroy abnormal cells, synthesize cytokines, and help mediate viral infection responses. How do we get so much diversity in our immune system? How is our immune system able to respond to so many different potential pathogens? If we look at our B and T cells, by the time they reach our lymphatic tissues, each one needs to be equipped to respond to a single unique antigen. This diversity in our T and B cells is generated by random shuffling and rearrangement of gene segments that code for the antigen receptors on both our T and B cells. If we had every possible recombination occurring, we would have an incredibly unrealistically large assortment of lymphocytes in our body. Our body wouldn't have enough room to store all of these lymphocytes. It's estimated that we have the potential of making 10 trillion different kinds of antibodies. Therefore, we have the ability to respond to 10 trillion different kinds of pathogens. So clearly, there's not enough room in our body to have this kind of response. So we'll produce some and keep some, but others that we produce, we destroy because we deem them unnecessary for our bodies. Another term for an antibody is an immunoglobulin. Immunoglobulin is a technical term for antibody. And an immunoglobulin, or antibody, is a large glycoprotein that serves as an antigen receptor on B cells. This immunoglobulin, when it's secreted to the extracellular environment and not on the surface of the cell, can also be referred to as an antibody. Generally speaking, most individuals use the term immunoglobulin and antibody interchangeably. All immunoglobulins or antibodies are going to have some common structures. They'll have an antigen binding site. This is the part that varies from antibody to antibody, and this is the variable region that changes. It is not the variable region that changes. It is the part that binds to our antigen. The variable region itself is going to change within the antigen binding site, and then we also have our light chain regions. These light chain regions, heavy chains, and constant regions are amino acids of our immunoglobulin that don't change very much from antibody to antibody. But these two areas do change quite a bit from antibody to antibody. So if we looked at our antibodies, we're first going to start with the genetics of our antibodies. And we can mix and match different sections of our DNA by changing how we edit the RNA. So we have randomized intron and exon editing as we process these pre-mRNA transcripts to produce processed or finalized messenger RNA molecules. And that's going to cause the binding site, which is up here. Let me fix this again. This whole triangular patch on top is known as the binding site. This is what binds to our antigen. And within the binding site, it's made of two variable regions that have both light chains and heavy chains. And then we also have the constant region at the bottom of the antibody. And this entire thing is known as an antibody or immunoglobulin, Ig. So the two variable regions will make up the binding site, and that is going to be opposite of the constant region of our antibodies. Our T cell receptors are going to belong in the same family as our B cell receptors. They're very similar to each other. They're formed by genetic recombination, just like the B cell receptors are. They have variable and constant regions. They're inserted into the membrane and have antigen binding sites. However, unlike B cell receptors, the T cell receptors are relatively small, and they're never going to be secreted from the cell. They'll never be free-floating in the extracellular environment. As our T and B cells are produced, we'll go through the process of clonal expansion and clonal selection. Clonal selection comes first. This is how we figure out which T cells and B cells belong in our body and which T and B cells should be reproduced and which T and B cells should be destroyed. This mechanism is going to help us determine exactly the correct T and B cells that can be activated by incoming antigens. Our lymphocyte specificity is going to be all pre-programmed. So once our lymphocytes are made, they are going to be able to respond to a specific antigen and only that specific antigen. And this response is pre-programmed into the cell before the antigen is present. If the cell is never exposed to the antigen, it is never going to be activated. Generally speaking, each distinct lymphocyte expresses only a single specificity and can react only to that one specific antigen. Clonal expansion occurs after we activate the lymphocyte, the T or B lymphocyte or T or B cell. So if we expose this T or B cell to the specific antigen that activates it, it'll go through clonal expansion and rapidly reproduce. So let's first talk about clonal selection. I think this figure does a great job of explaining it. We first have precursor or naive lymphocytes. Most of those lymphocytes are going to contain receptors on their surface that do not respond to self-proteins. Those receptors that are on their surface that don't respond to self-proteins are good. They mean that these cells, like this guy right here and this guy right here, can potentially help us fight off an infection later on in our lifetimes. However, we will always produce a couple lymphocytes that respond to self-proteins. And we want to destroy those ones immediately. If we destroy them right away, fantastic. They won't be reproduced. They won't be passed on as a mature lymphocyte. If we don't destroy them, they can be passed on as a mature lymphocyte and then initiate an autoimmune disease as they start to destroy our own tissues. So first we go through clonal selection and we make a bunch of different lymphocytes that can each respond to a different non-self protein or carbohydrate. And then later on in our lifetime, we may take one of those lymphocytes and expose it to the specific antigen that it's designed to respond to, and then that specific lymphocyte will rapidly reproduce and go through clonal expansion. So, concept check class. For each of the following properties listed, determine if it describes a T cell or a B cell. This is going to take a while for you to look up the answer to. You can check in your textbook, you can check in your notes if you printed them off, or you can rewind this PowerPoint video recording. So go ahead and pause it and get me the answer. 1, 2, 3, 4, 5. Okie dokie. If we look at option A here, option A is going to be associated with B cells. Option B is associated with T cells. Option C is associated with T cells. Option D is associated with B cells. And option E is associated with T cells. If you ever have any questions associated with this presentation, please feel free to shoot me an email, class, or post them in the discussion board. Happy studies.